spend a bit of time and be on your way again. So this is the outline of the talk. We're looking at the growth of bacteria and ways to monitor it non-invasively. And that leads us to the using a technology which is very well established and that uses tunable diode lasers. And we show that it's possible to measure the growth of bacteria non-invasively and with very, very high time resolution and with very high sensitivity. And we also tested this on a specific type of bacteria that emits light once the growth exceeds a threshold. And there's some interesting behavior that we've observed. And finally, we show you results of simultaneous emission of two gases by Cytobacter. As you can clearly see, these have implications as regards greenhouse emissions. Nitrous oxide is 200 times worse a greenhouse gas than carbon dioxide, and a lot of the chemical effluents that are dumped in the rivers is very nice food for the bacteria that then give off tremendous amounts of nitrous oxide. And so quantification of that under field conditions would be a useful thing from an environmental monitoring perspective. So we hope that we can do something about it. Now, the news about bacteria is not very good. We all know it. It affects us in many ways, even cancers. H. pylori, Heliobacter pylori, which infects about 80% of people in India, has been declared as a carcinogen of late. And detection of these gases in humans is also important, apart from the other infections that we know that there are. The antibiotics are meant to kill the bacterial cells without harming the human cells, but we've taken that a bit too much of antibiotics have been given out by the doctors, which has led to antibiotic resistance, which is growing very rapidly in the world. Drug discovery has been going down over the years. In the last 25 years, there's been no new drugs have been developed, and the emphasis is on prescribing less antibiotics so that the antibiotic resistance does not increase. It is projected that by 2050, large sections of populations will die because of very common things like common cold and cuts and burns, which will not be possible to treat because there are no antibiotics that can treat those kind of conditions. So the antibiotic catastrophe is something that is very real, and there's been a lot of activity all over the world in this direction. You can see that the percentage resistance has been going down in some of the countries that realized this problem early on, in the US. But however, in India, the trend is somewhat upward, and there's been examples of antibiotic resistance in neonatal cases, which is a huge worry. Then newborns are resistant to antibiotics, so there will be generations that will be growing up without any antibiotic, with antibiotic resistance. And you see the numbers over here are really alarming for India, more than double the rate in the US. So there's a lot of reason for us to worry about. This was a census done in Delhi, Mumbai, Vellore, and compared with South Africa. These are percentage resistance cases in Delhi, very high in Mumbai, and that's a result of generations of excessive antibiotic consumption by us. So there's something needs to be done about this, and there are lots of various different kinds of approaches that have been taken in measuring bacteria population. And the other step that has been taken is to move away from drugs into innovative things like graphene needles that are very effective in puncturing the bacteria cells and killing them effectively. So alternative treatments have to be developed, but the other thing that needs to be addressed is your ability to measure bacteria growth without invading it, without contaminating the sample, and fairly rapidly. So some of the most popular tests that are used to measure the quantity of bacteria takes as much as 16 to 48 hours depending on the bacteria strain. They're also invasive. You need to prepare the sample and the risk of contamination is high. So measuring bacteria growth is not as straightforward, and that is where delays in treatment have been shown. This is documented evidence that for every hour that you delay treatment, the mortality rate goes up by 9%, which is fairly high. So we started off this work trying to address this issue, whether we can get some rapid detection and whether we can get ultra-rapid detection of bacterial growth without compromising on these heads over here. So we realized that if we look at the bacteria growth curve, which is a very well-known process, these show distinct regions of growth. Initially, the bacteria lies dormant, it doesn't grow much, but then there's an exponential phase when it starts to grow. There's a lot of nutrients available to it, and slowly the death process also kicks in, so you have a balance between growth and death. And then once the nutrients are all consumed, then the death dominates. But we're mostly interested in this region where you have clear indication that the bacteria population has started to grow and exponentially. So if you could measure this part quickly, it would be an advantage. So I'll skip some of the mathematical details, but present you some of our results. The other interesting thing is bacteria don't just produce carbon dioxide, they produce a host of other gases in various quantities under various conditions. So depending on whether the respiration is in the presence of oxygen or in the absence of oxygen, in the presence of methane, in the presence of high pH, bacteria are very, very resistant. And so there's interest in various communities, not medical communities, people like astrobiology and other kind of environmental people. They want to see how bacteria grow under various kinds of physical and chemical conditions. So that's the other reason why we felt that detecting these gases would be an attractive way of measuring bacterial growth. And that's where we compare with the traditional techniques, gas chromatography and some of the other techniques have certain limitations, slow, bulky, you need sample preparation, you need to calibrate your measurements. So there are drawbacks with traditional measurement techniques. But that's where we thought if we could correlate this growth with the production of one of these gases, then that could be an indirect way of estimating whether the population has started to grow. So if the population grows and that amount goes up, then you have a handle on that. So we switch over to tunable diode laser spectroscopy, which is a fairly well-established technique of measuring gases and extracting information about their mole fraction, pressure, temperature, parameters like that. And with the advent of mid-infrared lasers, the fingerprint region has opened up significantly. You have very high sensitivity, you have wide tunable range, you can interrogate complex molecules, simple molecules, you can measure many molecules together. So this field has really opened up the possibilities. The mid-infrared is very rich in absorption lines. You would ideally want to zoom into one strong absorption line, which is isolated from other gases that may be present in the atmosphere, and you want to measure them rapidly and with high sensitivity. So it's important to select a proper line, absorption line, rotational vibration absorption line, so that you have zero cross sensitivity to other gases. So it is possible to pick such lines, and the technique basically is very simple. You have a laser whose current and temperature you can control, and you scan across an absorption line of a gas that is known to exist over there, and you get an absorption signal, and then you normalize it by the baseline, and you use a solid echelon over here to get a frequency scale, and you can superimpose this on that, and you can get an absorption profile, an absolute measurement, that's important, an absolute measurement as a function of the wavelength, and you can extract the concentration and the pressure of the gas from a single measurement. And you can do this for multiple gases, as many gases that your laser can access within a closed path length. It's not very sensitive. The problem with this technique is good because it's non-invasive, no sample preparation. This stop need not be removed. Once your bacteria grows over here, it puts out the gases into the headspace, and you don't disturb the experimental setup at all, and you can continue measuring non-invasively. So some of these are advantages, but the sensitivity is not that great. But the simplicity is very much there. So you have a laser over there, the bottle over here, and the detector. So experimentally, it's very, very simple. There's no chance of contamination, or stability is not that big a problem. Power requirements are low. So it's a useful thing. But for higher sensitivity, we need to move to a slightly different technique, which gives us much higher sensitivity. But I'm going to skip the details of this over here. Essentially, you add one more level of complication where you modulate the signal and you detect using a lock-in amplifier what happens to a modulated signal instead of a DC signal. So the technique is fairly well known to the sensing community. It is called wavelength modulation spectroscopy. You get much higher sensitivities, but your signals don't look like the absorption signal anymore. But you can relate this second harmonic signal to the absorption line. It is possible to relate that back, and you can get the same information out, which is the concentration of the mole fraction of the gas. So this is the technique that we use. We first use this on E. coli, and we were able to show last year that the carbon dioxide that is emitted by the sample of E. coli as a function of time up to 8-10 hours, that follows the same function as the growth population follows. So this gave us a very good way to measure relate the amount of CO2 that is produced to the population of bacteria that is present. And you can see from the signal levels, these are nice fits. So we apply the second approach also, which is more sensitive, and we are able to show then that the takeaway message for that is then you can monitor bacteria growth. You don't need to wait for 10 hours before you get a measurement. You get down to 3-4 hours depending on the bacteria strain, and you know whether the steps that you have taken to inhibit the growth are working or not. So the key thing for us is not tracing the entire curve. We would be looking at this part and further along over here. So the sooner you can make out that it has started to grow, the better it is for you. And it seems to, we are rather pleased that the tunable diode laser approach is working very nicely. So we went ahead, and this is the main work over here. Bacteria are also known to emit light, but they don't emit light until there is a sufficient quorum. So they signal, it's a mysterious thing, it's not very well known, but somehow it seems that they wait for a sufficient number of bacteria to be there before they all of a sudden switch on the light. And you can see it in a dark room, it's a green light. So we were interested in studying whether that behavior has any relationship with the carbon dioxide emission. And so we ran this test where we have a bacteria over here. The laser measures the gas in the headspace, and a single photon detector measures the light that is produced. And this experiment was done for about 20 hours, so that's my students' patients. And after 20 hours, we got very curious results, which we are not able to explain at this moment because it's very different from what's out there in the literature. So the first thing to note is the process runs for 16 hours, and the measurements are taken every minute. So this is the high resolution, time resolution aspect of it. So this shows you the photon count as time progresses. So this is expected that the bacteria waits till there's sufficient quorum, and then the bacteria starts to produce lots of light. So in the background, remember the growth is progressing. So there's initial growth, slow growth, then there's rapid growth, the exponentially increasing growth, and then there's a saturation of growth, and then there's a decay of growth. So it seems that as the bacteria start to grow, they realize we have a quorum. Now they very abruptly switch on all the light, so the photon count goes up. And then the growth begins to slow down because now the depth is starting to dominate, the substrate is being consumed. And so you would expect this curve to grow and then decay smoothly. However, it seems that there are many decisions that are being taken by the bacteria population. For instance, over here, the population is growing, but the amount of active bacteria perhaps is not as much. But then a point is reached again when the bacteria starts to switch on its light once again, and then it switches it back off and on and off. So this is very strange behavior. We don't have a theory for this. We don't understand enough of the biology, but it seems like a capacitor charging up and discharging very rapidly. So it's a kind of strange behavior. We repeated that once again. The results are not exactly the same, but there was a trend. So these trends are fairly prominent over here. The absolute values are a bit different. So we're still trying to figure out why this is happening. And then we superimpose all of those results, looking at the carbon dioxide and the bioluminescence from this bacteria, and we superimpose it over here. The blue line and the red, the blue line shows you the bioluminescence, the similar behavior. The carbon dioxide is the red one, and the fit is the green one. It turns out, and the dotted line over here shows you the specific growth. So the specific growth of the bacteria reaches a maximum value, and then it starts to decay. So this behavior is fairly well known, and it seems that the maximum bioluminescence coincides with the maximum specific growth curve. So it suggests probably that the bacteria decide after this that we are dying, and perhaps that's the reason why they reduce the bioluminescence. But again, we need to understand these much better. So that was that's a important experiment for us. I'll just quickly run through this video. If hopefully it works. How much time do I have? The 2 minutes? Sure. Right. So you see the bacteria. You see this is our 2F WMS signal from which we extract the mole fraction, and here the bioluminescence and the carbon dioxide content is shown. So the carbon dioxide goes up fairly monotonically, but the bioluminescence has started to go down, and we're still trying to figure out how much correlation the two of them have with one another. But we need to do more experiments, and doing 18-hour experiments is not trivial. So we'll have to persist with it a little bit. But we have the means to do it, and there's certainly the details we have to work out, but the overall behavior we understand. So the last bit, we looked at carbon dioxide and nitrous oxide from Cyclobacter, which is where we used a mid-IR quantum cascade laser, which is able to detect nitrous oxide, and we used a 2-micron VCSEL, which is aimed at a 2-micron carbon dioxide line. So there are two lasers in orthogonal to each other, and we have the sample in between. When we did this, this was again somewhat strange. The carbon dioxide follows the blue curve as before. However, the nitrous oxide, emission of nitrous oxide occurs at a different time, and the pattern is also different. So such results are not available. So we need to do more careful tests, but this is certainly showing us that the bacteria need not emit different gases simultaneously. And why that is the case, we don't have an answer at this point. We would look to work with microbiologists in future, study different strains of bacteria that are known to emit more than one gas, and see what kind of behavior we have. So in conclusion, we have a non-invasive method of giving us extremely high time resolution measurement of multiple gases that are being emitted by a gas. And finally, these measurements need not be done in the lab because the system is very small and portable. We can go to wetlands and dumping yards and wherever and make in situ measurements of processes that go on there. So that should give us lots of field data to work with in future. So that's where we are headed. That's all we have for you, and thanks very much to Photonics for the talk. You're welcome to visit us anytime you like. Thank you for your time.
Any question? What is the color of bioluminescence here?
That's about bluish green. It's around 490 nanometers.
Around 490.
We haven't been able to measure the spectrum because we don't have an OSA, but we'd be interested to look at the spectrum.
Spectrum and the bandwidth.
Yes.
Did we measure that?
No.
Peak is for around 490.
It's known to be about, for this one, it's known to be 492.
Right. So, the generation of gas is there, of course. But before that, we could monitor microscopically much faster growth of vaccine.
Actually, not.
No?
No. Because the density per square micron,
Okay.
that kind of density, it's a bit hard to do that repeatedly.
Oh, I got it.
And also, the other thing is, if you invade into the surroundings of the bacteria, its growth will be.
Yeah, so we can correlate that your tunable diode is a spectroscopy. What is the ppm level? You can go ppm level?
Yes.
Yes, that's the key point here.
So very low.
That is why that is the one. Otherwise, microscopy can also do.
Yes.
And that will be quite late.
Yes. But in you want to let the bacteria grow.
Yeah.
So from the microscope, you would interrupt the growth.
Yeah, but
which we want some
you could probe it up to ppm of 50 days. So
Yes.
that is much faster, right?
Yes.
Yes, correct. Thank you.
Are you completely sure of all the gases that are being generated? Could there be something else that's being generated that's
could.
absorbing
Yes.
and causing those fluctuations?
Yes, it could, because one of the things is the dissolved oxygen. This is aerobic growth. So the oxygen that's dissolved in the broth could be there. And currently we are not bubbling the oxygen. It's just the dissolved oxygen, whatever vapor pressure is on top. It could be oxygen-starved growth after a period, and then maybe it gets a bit of oxygen more. So there are lots of processes inside that could be happening. But now that we can measure what's happening on a minute by minute basis, we could we could model these differential equations, because the growths are differential equations. We could model these and include funny terms in the growth in the equation, and then solve it and see if it matches, and then try and rationalize why those funny terms should be there. But there's a lot of work that we can do, interesting stuff. And the speaker.
Okay.